So with regard to the chemistry that I was talking about, I think it's worth taking the time to kind of talk about the reactions that help with oxygen loading and unloading, and talk about them first independently from that process. Because once we kind of get into gas exchange and oxygen loading and unloading from hemoglobin, it's easy to get in the weeds really fast. So if we kind of parse it out and take it piece by piece, we can build the whole picture up towards the end and it'll be a lot more manageable. Plus, for a lot of you, it's probably been a while since you thought about enzymatic reactions and that kind of thing. So having a refresher is always a good idea. So RXN stands for reaction, by the way. And so let's first address what this means. So anytime I see the suffix ACE, A-S-E, I should automatically be thinking two things. One, that thing is a protein. And two, more specifically, that protein is an enzyme, which implies that it catalyzes some chemical reaction or another. And in doing so, it's not changed by the reaction. So the way it participates is to facilitate the reaction and then step away and be ready to do the same thing over again. So that's just a little memory jog for what enzymes are. The other thing that I want to remind everybody of before we get started is that there are some exceptions to this, but in biochemistry, there are very, very few. The overwhelming majority of biochemical reactions are reversible, meaning an enzyme can catalyze a reaction in one direction and also in the other. So we can have reactions that join monomers together to form a polymer, and we can have the opposite also be true, where polymers are broken down to form monomers. And which is happening is going to depend on the relative amount of reactants versus products. So we will address a little bit of that. But now, let's get into the nitty-gritty of this particular reaction, and we'll kind of analyze the pieces of it, and then we'll go apply it to what's happening in red blood cells and how that affects hemoglobin and oxygen. So there's our grand reaction. And I'm going to add a couple things. So I'm going to abbreviate carbonic anhydrase by saying Ca. And I'm going to draw that to indicate that in this reaction set, there's two halves, and carbonic anhydrase is responsible for catalyzing this thing specifically, and the double arrow indicates that it does so reversibly. So that's item number one to pay attention to. The other item to pay attention to is we're going to have to talk about acids and bases again, which is another concept from Biology 160 that you may not have thought of for a little while, so it's worth a refresher, right? So this is carbonic acid. And carbonic acid is a weak acid, which means that it does fit the definition of an acid. When it's dissolved in water, it gives up a proton to the solution, which is that H plus over there. But weak acids just don't give up their protons quite as readily as strong acids do. So adding one amount of carbonic acid to a solution is going to produce a smaller pH change to the solution than you would see if you added the same amount of a strong acid like hydrofluoric acid, which BT dubs is the strongest acid. You thought sulfuric was bad. Hydrofluoric will literally melt your face off. So how excited an acid is to get rid of its proton determines its strength. So that's thing number one. And thing number two is we actually don't need any enzyme at all to catalyze this part of the reaction, because this is just based on the way acids behave in water, which, you know, we're mostly water. The inside of cells is mostly water. All of the reactions I'm showing you today, or ever in this class really, are happening in an aqueous solution in water. So we've established that carbonic acid is an acid because it meets the criterion of giving up a proton to solution. And then it also has the conjugate base, which is the result of the dissociation of the acid, which this is bicarbonate, which is a weak base to carbonic acid's weak acid. So if I stick an acid in water, it's automatically going to dissociate, and you can mathematically describe the dissociation rate and a bunch of other things about how that works using something called the Arrhenius equation. So if you have taken or are going to take chemistry, that might sound familiar. We're not going to do the Arrhenius equation in here. I don't care about applying numbers to this. I mostly care that you understand how it works in specific contexts that are relevant to gas exchange. So we don't need an enzyme for this step because this is just what acids do, but we do see that this is a double arrow here. So that begs the question, how do I know whether it's going to be moving in this direction or this direction? And the answer to that comes in the form of equilibrium chemistry. So if a solution has a higher concentration of protons separately from bicarbonate, which we're going to say are the reactants, if it has more of those than the product, the net direction of the reaction is going to shift towards formation of the product. In the opposite case, if we have a higher concentration in the solution of the product, which is carbonic acid, and less of the two little reactants here, the reaction is going to shift towards the formation of more products. So it's a relative amounts thing. So we have a complex equation where one side is catalyzed by the reversible action of an enzyme, and the other side is quote unquote catalyzed, and I'm using that term very loosely, by simply the equilibrium chemistry of acids and their conjugate bases in a solution. So there's two things to worry about, which is why I wanted to parse this out separately for you before we sort of dive in and start applying this to things. So specifically as it relates to this side of the equation, we're going to see shifts according to relative amounts of things. But the outcome of that is really we're going to pay a lot of attention when we're worrying about gas loading and unloading and reaction chemistry. Our primary focus is actually going to be on how much carbon dioxide there is ambiently in that particular environment. because although the green half of the equation is catalyzed by carbonic anhydrase, it still obeys equilibrium rules. So if I have an environment that's really high in CO2, that's going to favor the conversion of CO2 into carbonic acid and thus the formation of a proton and some bicarbonate. If I have an area that's really low in CO2, that's going to favor a shift of the reaction in the opposite direction. So I'm emphasizing CO2 for a very specific reason, which may surprise some of you because I'm supposed to be talking about oxygen loading and unloading, not about CO2. And up to now, I haven't even really mentioned oxygen. So here's the cool part. What we are going to do, and say goodbye to this, is just take a moment once again to remind ourselves about mitochondria and what they do. Okay, so there's my crappy drawing of a mitochondrion. And what I should probably do is actually make it clear that this is a double membrane system. So I have one enclosed membrane space inside of another enclosed membrane space. So without zooming in too much and talking about things like the electron transport chain, we're just going to summarize what the mitochondrion does and what it does well. So in the matrix, which is the area that is inside of the inner mitochondrial membrane, we have things like the citric acid cycle and other stuff. So one component. And then the membrane reactions are where we actually use the oxygen. But regardless, what we're doing is we're trying to oxidize glucose because glucose has a lot of covalent bonds in it, and those bonds are full of energy. And that's the energy we want to extract. So if we oxidize glucose in a series of steps, we can really squeeze every last little bit of that potential energy out of the glucose. But in order to do that, we have to kind of play keep away with an electron. And electrons by themselves are pretty damaging. So they like to go rip apart things because they really want to be paired with another electron, and they also want to be associated with a proton. So if they're naked and allowed to sort of just operate independently, they go rip protons off of things and do all kinds of other nasty stuff. So we have to kind of control the behavior of electrons. So one way to do that is to just give it to oxygen, and that forms water. So what you end up getting is carbon dioxide and water, which is the lion's share of what we breathe out when we exhale. So you are breathing out a waste product, a gaseous waste product of metabolism. And when you give that electron to oxygen, you form what's called metabolic water, which is water that you make yourself from scratch. It didn't exist before, but now it does, which is pretty cool. You can make water. You do it all the time. So because we know this, and we know that all of our cells, for the most part, contain mitochondria, and they're all busily burning oxygen and glucose to make ATP because we have to have ATP made in accordance with its demand. If I look at a tissue that has really high metabolic activity, so one example would be, say, a skeletal muscle during a run or a workout, the corollary is that it's going to be producing a lot of CO2. So we can make a generalization from that, which really helps to kind of make the connection between CO2 and oxygen delivery, which I'll help you piece together in a moment. But through all of this, it's important to remember metabolically active tissues are going to release a lot of CO2 compared with tissues that are not so busy metabolically. So that's why, even though it seems a little bit counterintuitive to talk about oxygen delivery on the basis of CO2, CO2 production represents oxygen demand, and the chemistry links the two together. So let's look at that now. So what I'm going to draw is, well, I guess I'll draw a skeletal muscle. Since I use that as an example, why not? So I'm just going to draw a big, huge skeletal muscle cell. And I'm not going to draw all the striations because that would take 8 million years, and I don't want to draw any sarcomeres today, but what I will do is draw the super abundant mitochondria. And you also may recall that skeletal muscles have more than one nucleus. So they break the rule of one nucleus per cell. Okay. And then if I'm going to be a good professor, I should label this so that people who are not currently here know what the heck I'm talking about in the future. So skeletal muscle cell. So let's say, for the sake of argument, that I am using this skeletal muscle cell actively. I'm working out or something, which means that I'm going to be wanting to consume a lot of oxygen and pumping out a lot of CO2. So what we're going to start with is the pumping out of CO2. And I'm going to make like a really huge, artificially large blood vessel, just so that I have enough room to draw. So if this skeletal muscle is producing a lot of CO2, that means the intracellular concentration of dissolved CO2 is going to be high compared with the outside. And so that just sets up our diffusion gradient, where CO2 is going to diffuse down its concentration gradient out of the cell into the extracellular fluid and also into the plasma and whatever passing cells happen to be wandering by. I'm actually going to make this even bigger because I realize that I'm going to need a lot of room to draw this big giant red blood cell. I'm just going to label that as my blood vessel in case anybody was confused. And we're going to make the direction of flow be this way. And that's going to necessitate that I turn this purple and up here probably needs to be blue. So venous circulation. And then the purple capillary bed area. And then once we've picked up oxygen, it gets to be red. Oh no, I just wanted to erase that one little thing. Okay, so now that we've done that, I'm going to draw a very large erythrocyte because I need to leave myself enough space to draw all this stuff. And then I'm going to pause because we've talked a lot about CO2 and how metabolically active tissues produce a lot of it. We now have to address the other part of the equation. So I'm going to take a moment to pause and talk about that and draw a little bit about it in this little nook right here. So and if you're like, what are those blobs that you're drawing? If you're not, all will become clear momentarily. What color do I want to make this? I guess yellow. Okay, so I've just drawn you is an approximation of the space filling model of hemoglobin. Now, is it really orange and blue and yellow? No, but I have to have a way to illustrate the different parts of it. The other thing to know is this is not to scale. So what's really happening is that this red blood cell is just full of millions and millions and millions of molecules of hemoglobin. And that is what carries our oxygen. So hemoglobin is what's called a tetramer, which means it is consisting of 4 independent amino acid chains that are folded up and then glued together to each other. So we have alpha globin and beta globin making up the amino acid portions. And then there is a sort of flower-shaped pigment molecule in the center of all of those called heme. And at the center of the heme is an iron atom, Fe2+ specifically. So this is the thing that holds on to oxygen. So when you breathe in and you're gathering oxygen from your environment, it's not just dissolving into your plasma and cruising around in your plasma. That wouldn't be efficient enough alone to deliver oxygen to the cells and tissues that need it. We need a specialized carrier that knows where to drop the oxygen off and in what amount. And I'm using the word knows here loosely. Of course, it's just a protein. It doesn't know anything. But the chemistry that we're going to talk about is how oxygen drop off is able to match demand. So if this little guy is actually carrying an oxygen, each hemoglobin can carry oxygen, and specifically it can carry 4 of them. So 4 O2 or 8 total atoms of oxygen. So when O is bound to another O and it's in the form O2, we call it molecular oxygen because it's 2 oxygens bound to each other. And that's the form it takes in our environment and also in our blood. So basically the hemoglobin is carrying around, probably it's 4O2. And that's inside of this red blood cell. And this red blood cell is making the giant loop-de-loop of the systemic circuit. So the question is, hemoglobin is often abbreviated HB, by the way. So if you see me write that, that's what I mean. How does hemoglobin know where it's supposed to drop oxygen off? What tells it? It's just a carrier. So it's not going to bind permanently to oxygen. And if it did, none of us would be alive. So hemoglobin is kind of like an ice cream man. So a good ice cream man carries the ice cream to the children and then gives the ice cream to the children. The ice cream man does not hang on to the ice cream in perpetuity and drive around being like, I have ice cream, but you can't have it. Ice cream man drops off the ice cream. So same principle applies to our hemoglobin. We must have it bind to and pick up oxygen at the appropriate location, and it needs to hang on to that oxygen until it gets to the appropriate location to drop it off. But it's just a protein, it doesn't know anything, it's not alive. So then if that's true, how does oxygen drop off meet metabolic demand? It's a weird question. And for a long time, nobody knew because physiology was, we just didn't have sufficient resolving power. You can't see any of this with your naked eye, so you have to figure it out by experimentation. So, turns out the ambient CO2 that a metabolically active tissue is producing is the answer to how it knows, quote unquote. So I'm going to leave our little hemoglobin up over here just for a reference point. And then I'm also going to write a little fact over here. A decrease in pH leads to an increase in O2 popping off of hemoglobin. So the reaction chemistry is essentially such that it's shifts in pH that cause hemoglobin to hang on to or let go of oxygen. So let's examine this from the perspective of CO2 and our metabolically active tissue. So we know that it's going to diffuse from an area of high concentration to an area of low concentration, which means into our red blood cell. And we also know that, like all cells, the inside of a red blood cell is mostly water. And the inside of red blood cells also contains carbonic anhydrase. So if I have high ambient CO2, and I already have a lot of water because duh, that's going to mean that the relative amount of reactants, in this case, CO2 and water, is going to be high, which shifts the carbonic anhydrase reaction towards the formation of carbonic acid. And carbonic acid, because it's an acid, dissociates into a proton, which we denote as being H+. Hydrogen is 1 proton and 1 electron. So if hydrogen loses an electron and becomes positively charged, all it is a proton. So when you hear me say proton, but I'm writing H+, it's basically just two different ways to refer to the same thing. Hydrogen cation, proton, they are the same. And then it also, of course, dissociates into bicarbonate, which we will deal with in a minute. So we've already defined acids as being compounds that donate protons to solution when they're dissolved in water. And I also told you that a decrease in pH increases the rate at which oxygen is going to hop off of hemoglobin. So I'm going to draw a little mini version of my hemoglobin right here. So basically what this proton comes and does is it evicts oxygen. It's like, what's up? Get off of my spot, go away. And then the proton takes its place. So as CO2 enters a red blood cell, gets converted into carbonic acid and dissociates into its proton and the conjugate base, that means that the ambient pH inside of the red blood cell, as it passes the metabolically active tissue, the ambient pH goes down. The concentration of intracellular protons goes up. And whatever oxygen has been hanging on to hemoglobin all the way here has now been booted off of its spot. So it's no longer bound to hemoglobin. It is now free to diffuse from an area of high concentration because lots and lots of oxygen molecules are getting kicked off of hemoglobin in this particular cell at this particular spot. And it's going to diffuse down its concentration gradient as well. So the ambient oxygen inside of our skeletal muscle is going to be low because the second oxygen goes in here, it's getting sucked up and used by the mitochondria. And because we're having oxygen unloading happening at the red blood cell here, the ambient oxygen level inside of the red blood cell is going to be high. And we know that compounds, molecules, particles like to diffuse from an area of high concentration to an area of low concentration. So once this O2 is hopped off of its hemoglobin, it just goes right in here. So the cool thing about that is that the more metabolism a cell is doing, the more CO2 it will produce. And that will catalyze the following reaction in every single red blood cell it passes. And the amount of oxygen drop-off will be proportional to the amount of CO2 that that tissue is releasing. So due to this chemistry and in this way, we have this phenomenon where the supply of oxygen hopping off of hemoglobin is matched to the metabolic demand created by the cell or tissue that the red blood cell is passing by. So that's really cool. I would be remiss if I didn't talk about what's going on with our bicarbonate, however. So bicarbonate is a weak base, and it's really good as a buffer in a buffer system. So what a buffer system does inside of a solution is basically the buffer helps to absorb protons or bases and prevent large dramatic shifts in pH, turning them into smaller shifts in pH that are tolerated physiologically. So because of this minus here, bicarbonate can easily combine with any ambient protons to form a weak acid and then be converted back into water and CO2, which are both neutral pH-wise. So bicarbonate is a really powerful blood buffer for that reason, but it's not useful as a blood buffer unless it's in the plasma. So we have to get it out of the cell somehow. So it's not quite as simple as bicarbonate just diffusing out for two reasons. One, it's pretty big, it's a compound. Two, it's polar, and polar things have a hard time crossing membranes. Three, it's negatively charged. So the majority of our cells like to be more electronegative inside than their environment. So they like to have more minuses on their inside. And that charge separation helps to power lots of transport processes. So what we don't want to be doing is having our red blood cells hemorrhaging negative charge, because that's going to throw their charge separation all out of whack. So if I want this guy to go out here and be useful as a buffer, I have to trade it for something that's also negatively charged so that I don't lose my charge separation. So there is an exchanger that trades chloride ions, which are abundant in both the extracellular fluid and plasma, and trades them for bicarbonate. So that effectively pushes my bicarbonate out into the plasma where it's useful. And the absorption of a chloride ion basically prevents me from losing my charge separation because I'm exchanging a minus for a minus. So with regard to charge separation, cells don't care about the identity of the anion as long as they're trading a minus for a minus. So this process here is called the Chloride Shift. And it's also named after the guy who discovered it, whose last name is Hamburger. So it's also called the Hamburger Shift, which is quite funny. So by revisiting some reaction chemistry, and I was naughty and did not mention carbonic anhydrase catalyzing this. So let me put that in there. Based on this reaction chemistry, and the equilibrium reaction direction determinants. So we've got more CO2 in water favoring the production of more carbonic acid. We achieve the correct pH to encourage oxygen unloading that is commensurate with however much metabolism is going on in this cell. And we've also created a useful anion, bicarbonate, that we can stick in our plasma, and that helps our blood pH maintain steadiness, even if there's some metabolic problem that causes the pH to begin to drop. So bicarbonate helps us prevent acidosis of the blood. The chloride shift enables it to get out. And then the drop in pH that results from the dissociation of carbonic acid is what helps oxygen unloading to occur. So better living through chemistry, or in this case, just living through chemistry. If this didn't work, we would all be dead. So that is what happens at the cells and tissues. And I'm going to pause recording for a moment. Resume recording. Okay, so we're back on. So now we are going to look at the same sets of reactions and events, but in reverse at our alveolus, where the relative concentrations of oxygen and carbon dioxide are reverses. So let me just erase this first. Goodbye. And I'm going to draw myself a slightly more detailed alveolus because I think drawing the respiratory membrane. Why did I put the nucleus there? That was so stupid. Okay, let's put the nucleus up here so it's like out of my way. And then we just have a super thin little bit of cell. And then here's its neighbor. And then over here we have the endothelial cells making up our little alveolar capillary. So everything is very, very thin, which is good because that means that this, our diffusion distance, is very tiny, and that's going to facilitate good gas exchange. So over here, I'm going to do some labeling. And because alveolar air is ultimately connected to atmospheric air, we're going to have a lot of nitrogen. Because atmospheric gas is mostly nitrogen, which normally isn't a problem unless you decide to go very high up in the air or you decide to go very deep into the sea. Then it becomes a problem. We can talk about the bends later, not in this one. Okay, so we have very high ambient O2 and nitrogen. Should probably draw some more nitrogens, but I'm not going to worry about it right now. And I have relatively low CO2 because it should be environmentally low. Okay, so that's my alveolus. This is just the gas mixture that I've successfully extracted from the environment. And let's add some hydrogen in there just for good measure. Okay, so that's alveolar air. And then I have my alveolar capillary. And what we are going to do is go ahead and draw a big, huge erythrocyte passing through. Okay, so fortunately, now that I did the work already to explain the reaction chemistry and talk about oxygen unloading and sort of go through all the nuances and details there, it'll be easier to do this because essentially at the alveolus, everything I just showed you that happens at the tissue happens in reverse. So there's no added fanciness or anything necessary. It's just because the chemical reactions are equilibrium reactions, where the direction the reaction proceeds in depends on the relative amounts of reactants versus products. If you set up the right concentration gradient, the thing runs itself, which is fantastic. So as you can see here, I have high ambient O2 in my alveolar air, and in my blood cell, which has just returned from dropping off oxygen, I have really low ambient O2. So that's just going to set up a nice concentration gradient that naturally just obeys the laws of diffusion. But that doesn't solve the problem of getting that proton off of hemoglobin and having O2 replace it. So what we're also going to have is bicarbonate has entered the chat. So the hamburger shift goes in reverse.
Alexa, stop.
At the alveolus. And we get to kick this chloride ion out. So again, just happening in reverse. So if we want to trade anions around, we just have to make sure that we are trading minuses for minuses in the appropriate amount so that we're not losing or gaining too much negative charge, keeping that nice and steady. Okay, so once our bicarbonate is back in, we can combine it with our protons. So now I have to draw hemoglobin. I'm actually going to make, no, red's going to be confusing. What color should I make you? How about neon green? Sure, why not? So my protons have been riding around on the heme group of hemoglobin. But what that creates is a situation where the ambient pH inside of our red blood cell is pretty high, because when a proton is bound to another compound, it can't contribute to the pH of a solution. So when that's the case, that changes the affinity or the willingness to bind of hemoglobin. And as long as there's more oxygen ambiently coming in, oxygen will now be booting the proton off and taking its place. So once that proton is off, it is free to combine with our little bicarbonate here. And via the action of our friend, the Arrhenius equation, and just the rules that acids and bases follow in solution, if we have more of these separately, the tendency is going to be for them to combine to form carbonic acid. And you might be wondering, well, if these things are really abundant and they keep combining, then wouldn't the amount of carbonic acid just keep going up and up and up until the reaction reached equilibrium and no more was formed? And the answer is, it doesn't. Even though carbonic acid is consistently being formed inside of this cell as it's making its way past the alveolus, it's being dissociated as fast as it's forming because carbonic anhydrase is turning it from H2CO3 into water and carbon dioxide. So it doesn't stay in this form for long enough to contribute to a concentration change. So even though it's forming, it exists for the blink of an eye, and then it's catalyzed by carbonic anhydrase rapidly into CO2 and water. And so that keeps the reaction moving in this direction. So what that creates is a scenario where we have very, very high ambient CO2 inside of our red blood cell, and there's relatively low ambient CO2 in alveolar air. So after that, diffusion takes over, and we just dump that CO2 right back into the alveolar air, and we breathe it out. So, like I said, all that stuff is just happening in reverse. And the reason that it proceeds in the reverse direction is, again, because these are equilibrium reactions where which way it's proceeding is going to depend on the relative amount of reactants and products and a couple other things like concentration gradients and ambient pH. So that is oxygen loading and unloading and the chemistry that determines it. So as long as you have a small diffusion distance, you have sufficient hemoglobin, which includes sufficient iron, and you have enough red blood cells, you can effectively exchange gas and you can keep your oxygen supply matching the demand metabolically of the cells and tissues that require it. And that matching is very close. So that concludes the fun and cool and also very busy chemistry of gas exchange.